So today I'll be discussing some of the results from my lab. The work involves studying plastic degradation using beetle larvae. So we are trying to use mealworms and superworms to see if they eat plastics and the plastics indeed in their gut it's actually being degraded or not. So uh, my experimental setup was pretty much diet normalization treatment, then the genomic DNA isolation, gene abundance analysis, and then identification of the enzymes of interest. Uh, for my methodology, we first analyze the alpha beta diversity, followed by MAG analysis, and then the metabolic analysis using the read data, and then trying to find out what are the enzymes which are actually involved in the degradation of plastics, and discovering any kind of novel enzymes that may be involved in plastic degradation. So for the alpha diversity, we found some of the genera group which were abundant. Using the redundancy analysis and the feature data, we were able to see what kind of genera is actually pushing towards what kind of diet, or what diet is actually promoting what kind of genus group. The beta diversity showed us a clear indication between the gut microbiome of both the larvae, the mealworm and the superworm. There was a clear distinction, but the PCA did not give us that much information. Both PCA and the NMDS graph, we did not see like a good cluster of diets, like which could suggest that there is a definite microbiome belonging to a particular kind of diet. Then we use the MAG data analysis to see what kind of MAGs are prevalent in what kind of diet, and we were able to see some MAGs which were significantly found more with respect to standard diet and styrofoam diet. So we had like two different kinds of plastics, polyethylene and styrofoam, and we had a standard diet and a normal diet. Now, using the co-occurrence network, we try to correlate between what are the MAGs that cluster together, and then try to correlate it with the metabolic pathways. So as to see like which MAGs actually might be involved in plastic degradation. For both superworms and mealworms, it is yet to be conclusive, like what kind of metabolic pathway is actually predominant in these MACs. Our team is also sequencing almost 1,000 genomes of different bacterial species that were found in the experiment throughout the. It's like a very long project. It's been going on for like more than a year now. Now my second phase of the analysis was to understand like if there is any functional lackases that can indeed have uh, 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 the tendency to degrade the plastic. So for this we took two approaches. First was sequence analysis, and next was structural analysis. So for sequence analysis, we use sequence similarity, simple amino acid based similarity, to see what kind of enzymes are clustered together. So we already have some enzymes which have shown PE degrading activity. So using that, we did our clustering analysis, and as the threshold increase, as you can see, like the clusters also increase. But it also shows like there's a lot of diversity between the lackases that we got from our metagenomes. It is not conclusive that we are yet to understand like if there is a particular group of lackases that can degrade plastics, or indeed there is like a diverse group of lackases, or are there any functional motives. With that intention in mind, we wanted to do the structural analysis also. So for structure based similarity analysis, we took TM-align and fold-seq, and we took the data from it, and then we did GMM clustering, spectral clustering, to see how much the lackases that we got from our metagenomes are similar with the lackases that has shown PE activity. Now, the lackases that have shown PE activity also does not cluster together, which means that there might be a diverse group of lackases that might be involved in PE degradation. Uh, yeah, it's the same thing. So now from the results that we got from the previous step, we wanted to converge even more towards the binding site. So we now know the mags, some of the mags are involved. We know that there are lackases that are involved, but exactly what are the sites, or what are the characteristic feature of these sites, of these lackases, that makes it so makes it unique in a way to degrade plastic or degrade polyethylene or polystyrene. There are studies which have shown that there are residues of interest in the active site region of lackases, which makes it more functional. So with that information in mind, we did some statistical analysis, and our hypothesis was uh, the substrate comes and binds to these laccases, and then there are these important. So laccase is a multi-copper oxidase, so it has these important amino acid residues through which the copper is bound, and then we wanted to find these residues so that we can. So our hypothesis was to see if these residues or their distribution can be used as a criteria to see if these are indeed the laccases that have PE degrading tendencies. So we did different distribution analysis and found out like cysteine, histidine, isoleucine indeed showed like a differential abundance throughout our laccases. We had around 450 laccases we were trying to understand, and as of now we have like a group of laccases, and we are trying to find out like which of these laccases have the PE or PS degrading um, characteristics. So, at the end, I would like to thank my supervisor, Dr. George, and all my lab members and all my funding agencies for this work. Thank you so much.